Thank you very much. And thank you for the organising committee for allowing me to present today. And I'm going to follow on a similar theme from our invited speaker. So I thank you so much, Associate Professor Cheng, for introducing the topic. But we're going to talk about it from Grapevine perspective. And is it moving? Yes. So just to set the scene a little bit, because Grapevine is very old, personally I think very important, but very interesting crop in that it has a number of features that impacts its breeding quite significantly. And one that I want to point out here is around the clonal propagation. And you'll see a little bit later about why that's so important, but ultimately this results in a lot of, well, we want to do it to maintain our high heterozygosity, which is super important in Grapevine, but it also, as a result, creates somatic mutations and epigenetic changes. Some other features around Grapevine is its regenerative growth, meaning that it regenerates every year in the same position. So experimental design is very important because it will be there for a long time. But ultimately, all of those things come together, and Grapevine breeding has an extremely long cycle time, up to 25 years in some context. So all of these predictive modelling approaches that we've been talking about today and we'll go on to talk about in the conference are really applicable and have a lot of value in a perennial like Grapevine. I'm going to talk about what I would consider more of an emerging approach, which is multi-omic predictive modelling. And as we heard from the previous speaker, this is about all the intermediary layers. So, you know, we've got our really stable genome, and then we're moving through all those more flexible fluid layers to get to our phenotype or phenome. And it's about the interactions between those layers and also their interactions with the environment and the management, how we can pull those together in some kind of crazy model across traits to better predict our phenotypes. So this is what we're really interested in, but we're coming at it probably from a slightly different perspective, that we're looking at it as a tool for biological insight into the genome to phenome interactions. So today I want to share with you our case study. And so this is in a subset of clones. So it's not super clear, but up on your top left-hand corner, these are all technically the same genotype. And phenotypically, you can see that the clone is not a clone here. When we look at it from a genetic point of view in terms of our SNP genetic profiles, we're almost capturing a million variants. In terms of differentially methylated regions, we're capturing 450,000 of these kind of epigenomic features. And so this is really interesting from our crops perspective. But what we wanted to do is make use of this data, and I must just say I'm picking the right side in my presentation today. Oh no, sorry, it's not the right side, but yes, the right side of the equation. And so what we want to do with this data is create it into some relationship matrices. We've looked at these in a number of different ways, and unfortunately I don't have time to go into it today, but happy to talk about it later. But ultimately we're creating them or generating them so that they are contributing unique information into our model. We then need to combine this with some really good and nice phenotype data, and we're lucky that we have 13 years across 4 fields at our Geisenheim vineyard, where we've sampled 5 really important traits for grapevine breeding. And so what we want to do is have a very kind of first look at how we might start to think about building these kind of multi-omic models. And we started really simple, and I should just say this whole presentation is quite new work, and our whole perspective is just to start simple initially and build on to more sophistication. So we're playing around here with 3 different models, and we've got kind of our baseline GBUP model here, where you can see we're modelling the clone effect as random, and we are proportional with the variance proportional to our GRM. Then for the blue value here, this is where we then fit the ERM component of that, and then in the final green model here, we're fitting also some different non-additive matrices depending on the trait, but I won't go too much into that. But ultimately, what you can see here is that if we look at everything relative to the baseline of the GBLUP model that we are capturing a little bit of extra genetic variance from a narrow sense heritability perspective. But what I'm really interested in is, okay, so what of those omic features are actually contributing to that increased capture of total genetic variance? And so what you can see here is straight away, our blue colour here is our epigenomic features. And so focusing in on the bars on the right-hand side here, which is our multi-kernel models, you can see that the epigenomics is swamping across a lot of our traits, and we are getting a little bit of non-additive variance there as well. And so this is pretty exciting for us in the Grapevine space. We had some ideas around this earlier, but this is probably the first time we've seen it on this kind of larger predictive modelling approach. But this is, I suppose, we're talking about total genetic variance here, so we do need to take a step back. And if we look at it proportional to total variance, we start to look at this across some of our more environmentally sensitive traits like our acids. Doesn't look too great in terms of, you know, we've got a huge amount of environmental variance going on there. And as I said, this is a very simple model. So it's likely that our E here that we're capturing is masking some underlying G by E interactions that we haven't modelled in this framework. So obviously, the next step is to look at a environment by omic interaction modelling approach. And here you can see the equation. And again, this is kind of a first look that we're going with, and I'll just briefly run through it. So we've fit this to two different traits. Oh, looks like the boxes have moved a little bit, but you get the gist. So fixed effects, we've got intercept, field, and year. We've got our random year by omic effects. And here, so basically, again, the clone effect is proportional to either, you know, your GRM or your ERM, depending on the kernel. But we're modelling the variance covariance matrix as an unstructured variance structure. We then have our random field by omic effect, and here we've actually just fit independent variance covariance structure, and similar for a random field and year interaction. And then obviously we have our plot level residual, where we are modelling an individual variance for each year for each plot, but we're modelling that as independent as well, and there's a reason for that. So yes, that's kind of the base kind of model that we're looking at when we're trying to think about these environment by omic interactions. And we've modelled this for two trait examples, and if we first have a look at yield, you can see the variance components here. And straight away, you know, before we had about 75% of the variance coming from our year and a residual component, and now we've shrunken that significantly to under 8%. And overwhelmingly, we can see there's a huge amount of variance that's coming from this year by epigenomic interaction. And there's a little bit also coming from the genomic interaction there, but not so much from our fields. So that's yield. We also wanted to look at another quite different trait, and this is tartaric acid. So this is the stable form of acid in the juice, and it's quite important for winemaking. And you can see a slightly different story here. So, and if you remember from the previous slide, this trait, about 90% of the variance was coming from environment and residual. So we've done a bit of a better job by fitting this G by E model. But again, we can see that our ERM is dominating in terms of the genetic variance, but not so dramatically compared to yield. And we're still capturing a bit more from our GRM there as well. So I think that was really interesting to start to pull out some of these variance components from this model. We then had a look, obviously, calculating the genetic correlations from this model. We had a little bit of look at, I suppose, the patterning of the G by E across the years, or I suppose looking at how the clones are performing. And you can see when we estimate, or we generate these EBLUPs from the GRM kernel, that we're probably getting signals that are suggestive of crossover G by E. But when we do it from the epigenomics, we see that we're getting something that's a little bit more closer to maybe scale G by E. So we've got less re-ranking, and the clones are changing more together over the years. So that's the yield situation. And then if we look at tartaric acid, again, a different story, which is very interesting. But here, we see strong year by crossover G by E interactions. And, you know, as I mentioned, tartaric acid is a very environmentally sensitive trait, so there could be some reasoning there biologically as well. So, and then another thing I should have mentioned is that these clones were actually selected many, many years ago from nurseries across Germany, France, Austria, Luxembourg, and so they come from different regions, and then they were all brought to Geisenheim and clonally propagated. But when they were selected in those original vineyards, they were selected on agronomic traits. So we do expect to see some differences between our agronomic traits versus some of our juice quality traits here as well. And so I suppose the next step in coming back to our point of really starting to look at the biology and how we can use these models to look at that biology is, again, this is a very first look, and so it's very simplistic, and we would like to build on this with a little bit more sophistication. But what we did, and I'm just using yield here as an example, is basically from these EBLUPs that were generated separately from the GRM interaction versus the ERM interaction, we back-solved basically the feature effects for these. So we managed to get marker effects for each of the omics, and then what we did is basically we took the genome and we cut it into equal portions, just under 3,000, to create these genomic windows. Then within a genomic window, I'm just really interested in is how do these windows change over years? And so here we just calculated a very simplistic stability course statistic where we basically looked at the mean effect divided by its standard deviation. And so I think straight away it's just interesting to see that there's a big difference between the signals that we're getting between these omics layers, and you know, you can see that we're getting much more effect coming from our epigenomic features, and that their stability is varying a lot more when we look at these, I suppose, higher effect windows. And I think then comes the next question around, well, how related are these omics features? So I should have mentioned earlier, we orthogonalized the relationship, the epigenomic relationship matrix against the genomic relationship matrix. So, you know, then how related are the signals that we're getting? And so here we've also plotted the stability scores that were coming from the left-hand side of this graph against each other. And so obviously, ideally, you want something, if you're looking for a stable signal across years, a stable window, you're looking for something in this top right-hand corner. And then what we've done here is looked at really basic similarity using cosine, just to see, you know, how much these kind of windows are changing temporally, scale-free over time. And you can see that actually for this kind of top right-hand corner that we have a bit of an inverse relationship between with these two omic features, and then we have some more in the moderate space that are somewhat a bit more consistent. So I suppose the point by showing that little bit of extra example there is that we can use this to really start to delve into the biological insight. And what we want to do next in this space is really start to do a little bit more sophistication in some of the models that we're approaching, some of the metrics that we're using. But it's really about then, you know, under those windows, under those haplotypes, what are some of the genome features that are influencing the trait or the interactions that the omics are having with the environment, and then, you know, understanding those that environment through environmental covariates, but also adding omics layers. So here I've only spoken about 2 omics layers, but also we want to build that out to other phenomics layers like phenomics that we have a lot of hyperspectral data for, and then obviously build across different models to generate more biological insight. So yes, I just wanted to thank you all again and acknowledge all of my collaborators and the Department of Plant Breeding at Geisenheim and our sponsors.